So hi, good afternoon. My name is Victoria Lara-Aguilar, and for my Keystone project, I'm presenting my engineering senior design project, where I, along with my two other team members, developed a midbrain organoid-on-a-chip for live cell imaging and Parkinson's disease modeling. So Parkinson's disease, or PD, affects 1 million people in the US and 10 million people worldwide. And one of the reasons why there's no cure for the disease and the cause remains unknown is the lack of accurate models, as current animal and 2D models don't really translate to human models or capture cell interaction and organization. What researchers have found, however, is that the disease affects the dopaminergic neurons in the midbrain, and symptoms start showing when 80% of dopaminergic neurons are dead. So by that point, treatment options can only do so much. Hence, our proposed solution was the midbrain organoid-on-a-chip, or MOC, which I will be calling it, which will hopefully facilitate the cell growth and imaging process so we can learn more about PD cell dynamics. Just a little background information, organoids are self-organized 3D tissue cultures derived from stem cells that essentially make a miniaturized organ. Some of the benefits this MOC will provide are that compared to other similar protocols, the stem cells we will be using are human neuroepithelial stem cells, which differentiate into the midbrain a bit more efficiently than other stem cell types. The protocol that we are following is designed for imaging with an inverted microscope, which happens to be far more expensive and require additional training. So we're adapting this protocol so that it's compatible with an upright microscope, since they tend to be more common and accessible in labs. And hopefully this encourages other labs to replicate our design and also contribute to the research. Additionally, another benefit is that by embedding the organoids in the way that we're doing it, we should expect better images than other protocols that have the organoids either free-flowing or squished against a cover slip. And finally, the design can hold up to 9 organoids, so we can image whole and multiple organoids in a single session, whereas other designs slice the organoids, which again, don't really paint the full picture of what we want to know. So we tackled this project with 2 aims. So for the first aim, we focused on designing and assembling the chip and culturing the cells. This is the original protocol that we're following. And essentially on the outside of the Petri dish, we glue down a membrane for the middle 9 holes right here. And then we cure it with a UV light and place it in an incubator for 2 hours. Then someone places the cells on top, and then the glass cover slip is sealed on top, which is this square right here. And that prevents, that's just sealed, and it prevents the microscope itself from touching the cells. And then later we add Matrigel, which kind of holds the cells in place and prevents them from flowing around everywhere. So as you can see in the original design, the Petri dish is face down, so the media fills up the Petri dish on top, and that's where the design is built to be imaged from below. So we started thinking of some possible designs, and we came up with these 4. For A, it's basically the same thing as the original design, except that we seal it with some parafilm to prevent leaks. We flip it for the imaging session and then flip it back and remove the seal when the imaging session is done. For B, we dump out the media and flip for imaging, but as you can imagine, that wastes a lot of media and it can potentially damage the equipment if there's some media left over. For C, it's very similar to the original design, but instead of being on the outside of the Petri dish, it's on the inside of the Petri dish, but the procedure remains the same. And the cool thing about this one is that you minimize dish manipulation, and the chip remains in the same orientation throughout its imaging session and just its normal maturation process. D is a little bit more complicated, and it resembles a salt shaker in a way, in that you can close the holes and leave the media in the upper reservoir, but if you rotate it, you can open the holes and then it empties onto the secondary reservoir on the bottom. So with this as our starting point, we developed 6 main design criteria, and really focusing on sterility and constant media flow to the organoids and making sure that they stay alive. So we try to weigh our options before prototyping to save time and money, and we came up with this Pew matrix where we ranked how each design fit our criteria, and we find that A and C fit the best, as you can see there. So since I was primarily involved in the design, device design and assembly, here are some of the preliminary experiments that I did. So this first picture is a practice assembly I did to grow comfortable with the protocol and kind of fine tune some UV curing times. The second picture is a water test where I filled the dish with media. I sealed it, flipped it, and left it on for 72 hours, and there was no leaking, so we went ahead and improved that design. And then finally, this third picture was to prove if the inverted design would allow for the media to travel upwards and provide nutrients for the organoids continuously. And so we laid down a membrane and the media still diffused to the Petri dish that would hold the organoids. So we approved that one as well. For AIM 2, we focused on fine-tuning our imaging techniques in order to characterize healthy cell networks. So this is what the mock looked originally after assembly. And so we infected these cells with GFP, which is essentially a virus that's going to enter the cells. And when they're alive, they're going to glow, kind of like this, we can see. So this is the membrane, and these are the cell clustered together. And as you can see, they're pretty bright, and they form like a bright little circle. Now, this is a week after. As you can see, they're kind of scary looking and they're not ideal. So this is a membrane and this is a weird blob that's not really shining. Same here. There's some tears in the membrane over here. There's an air bubble, some fibers. So not where we want it to be. So this forced us to look a harder look at what we were doing, find out what was going wrong and how we could fix it, and just improve on the way we carried out things. So this troubleshooting actually led us to find that the UV cured glue wasn't cured for long enough, and that it was actually poisoning the cells as they continue to grow. And after that, we kind of retraced all of our steps, and then we focused more on sterility and efficiency as a whole, and actually ended up increasing the curing time more than threefold. So these are some of the results that we got after the troubleshooting. This first one kind of served as our control. This is the squishing that I was talking about. Essentially, it's placing a cell between two glass cover slips and lodging the cell between those. And while the picture is pretty clear, we can see the bright blobs right there. It's not the best in terms of what we want to find. We can't continue imaging since it kills the cell, and the shape just becomes distorted. Instead of being a circle, it's kind of like a weird blob right there. This is what we got with design C. So we can see some bright flecks, bright dots. So the cells are pretty much alive and they're doing well. However, we do see this cloud of autofluorescence, which is essentially noise caused by the media itself. So we can see that in both of these mocks. And finally, design A. Again, we see the autofluorescence kind of create some background noise, and we are able to see some of the cells, but they're not as clear as the ones in design A. So based on what we have so far, we can say that design C was our winner. So future direction, overall, we hope to continue troubleshooting. We believe that the background noise, the autofluorescence that I was talking about, is caused by the dye itself in the media. So we plan on removing that dye for future mocks as well. We're planning on making the organoid smaller to ensure that they get enough nutrients and kind of to avoid overcrowding and squishing within the device itself. And based on some mocks that we've made recently with these precautions, and we've been able to take a peek at them, we think that this is leading us in the right direction. If we've been getting some pretty good results, but those mocks are still pretty young and they need to mature a little bit further before we image them. And because of the troubleshooting, we unfortunately never made it to the Parkinson's disease part, but we hope that these future mocks are exposed to MPB+, which is a neurotoxin that will induce these Parkinsonian symptoms. And based on this, we should be able to learn more about these disease networks and hopefully bring us one step closer to seeing how PD works and eventually find a cure. Just a special thanks to everyone at the Han Lab, Shui Han, our advisor, and our two grad students that have been super helpful, Emma Bortz and Andrew Martin. Any questions?